Hello, everyone. I would like to welcome you to today's webinar, Unveiling the Dynamics of SARS-CoV-2 Variant Evolution and Its Implications. My name is Lucy Sue. I'm a Senior Product Manager at Twist Bioscience. I'd like to give you a few housekeeping guidelines before we get started. All lines will be muted during the webinar. If you have a question, please submit it in the Q&A box rather than the chat box, and we will answer all your questions after the presentation. If you share your name, we'll be able to get back to you after the webinar if your questions have not been answered. Following this presentation, if you have 1 minute to take a brief survey, we would appreciate your feedback so that we can continue to improve. The survey will be open automatically in your web browser. I'm very excited to introduce you to our speaker today. Dr. Tyler Starr is an assistant professor in the Department of Biochemistry at the University of Utah. Dr. Starr received his PhD in biochemistry and molecular biophysics from the University of Chicago, where he studied protein evolution in the lab of Dr. Joe Thornton. He then moved to the Fred Hutchinson's Cancer Center to conduct postdoctoral research on viral evolution in the lab of Dr. Jesse Bloom. Dr. Starr's research explores the evolutionary arms race between viruses and host factors involved in infection and immunity. Dr. Starr uses high-throughput deep mutational scanning assays to characterize the protein-protein interactions between viral glycoproteins, host receptors, and antiviral antibodies. These experiments revealed the biophysical details of protein sequence, structure, function relationships, and provide maps for understanding viral evolution and antibody development. Since 2020, Dr. Starr has applied these approaches to study SARS-CoV-2, informing ongoing efforts in viral forecasting and aiding in the development of vaccines and monoclonal antibodies to combat COVID-19. His lab is continuing to extend these approaches to understand the deeper molecular evolutionary features that drive the emergence of animal viruses that spill over into humans, and to inform the development of vaccines and antibodies that can treat or prevent future viral spillovers. And with that, I'll hand it off to Dr. Starr.
All right, thanks, Lucy. Let me get this set. All right. Thanks everyone for joining. And sorry, I just need to get this little Zoom control moved out of my view. All right. So hi everyone, I'm excited to tell you about some of the work we've been doing, kind of leveraging advances in DNA synthesis to make these big libraries to allow us to, at scale, characterize molecular evolution of SARS-CoV-2 and some of its related background viruses. So big picture intro, as Lucy already said, kind of the interests of my research in my lab is studying protein evolution at the host virus interface. And in particular, we focus a lot on these viral surface spike proteins, which are the proteins these viruses use to bind to receptors on host target cells. And it's this interaction between these viral spike proteins and receptors that allows the virus to enter into a cell to initiate an infection. And these viral entry proteins, like SARS-CoV-2 spike, are also dominant targets of aspects of immunity, such as neutralizing antibodies, which bind to this protein and inhibit its ability to interact with receptors. And so because of the importance of these kind of forward and chase and escape interactions at the host virus interface, a lot of really interesting molecular evolution goes on, both in this viral protein as well as the host receptors and the host antibodies that it interacts with. So to kind of think about these interactions and their evolution, I typically think in terms of fitness landscapes, kind of a classic framework in evolutionary biology, where we have, a space of many possible genotypes that any of these proteins can occupy. And at some point in time, any given gene exists in a certain sequence with some fitness that it confers. So what we try to do with our experiments is perform these high throughput demutational scanning experiments to basically survey the sequence space around proteins of interest with respect to different properties, such that when we then compare to what actually happens during viral evolution, so we see where the virus actually goes, we can compare it to our kind of mutational scanning maps to understand what were the factors that were driving the evolutionary trajectories of the virus or the host receptor or antibodies. So today I'm going to be telling you about a lot of the work we've been doing in this space focused on coronaviruses, and in particular SARS-CoV-2, whose viral surface protein, as we all know, is this spike glycoprotein. And in particular, we've been focusing on a small domain of the spike protein called the receptor binding domain, or also referred to it the RBD. This RBD is the part of spike that actually binds to host target receptors, such as ACE2 on the host target cells. And that's what triggers the kind of downstream conformational changes in spike that lead to fusion of viral and host membranes, and again, the initiation of infection. So therefore, this interaction between RBDs and ACE2s is really important in the evolution of SARS-CoV-2 and its sort of deeper bat coronavirus progenitors. So for example, it's known that evolution of RBDs to bind to different species ACE2 orthologs is obviously very important for things like cross-species transmission and zoonosis. And the RBD is also the dominant target of neutralizing antibodies, for example, in all of our bodies as a result of vaccination or infection, as well as all of the monoclonal antibodies that were authorized for clinical usage target this receptor binding domain in particular. And so because this RBD is kind of at the center of these different arms races, we've seen a lot of variation in the RBD over evolutionary time. So over the sort of what I'm going to call short-term evolution of SARS-CoV-2, as this pandemic has progressed, we've all kind of seen these variants of concern emerging, which fix many mutations across the viral genome, but in particular, they concentrate a lot of mutations in the RBD that impact these traits of ACE2 binding and antibody escape. But also, if we kind of zoom out to a larger sort of phylogenetic perspective on the kind of family of related bat coronaviruses from which SARS-CoV-2 is a particular branch that's spilled over into humans, over the sort of longer term evolution of these bat coronaviruses, this RBD is one of the most variable domains, again, because of these arms races that also exist within these natural bat reservoirs. However, sort of at both of these scales of evolution, we've become really good at seeing genetic variation. So we can sequence millions of genomes of SARS-CoV-2 worldwide, or we can isolate or sequence from metagenomic samples viruses out there in nature. But what sort of lags behind the sort of seeing of this genetic variation is understanding what the functional consequences of genetic variants mean, right? There's many more mutations occurring in SARS-CoV-2 than can be queried through traditional lab assays. And so typically when you see a new SARS-CoV-2 genome with a new mutation, you don't always know what it means. So what we've been doing to try to fill this gap is to perform kind of more prospective, high throughput biochemical measurements where we can characterize the impacts of mutations on key properties of these proteins prior to their actually occurring in nature, such that when a new variant of concern emerges, we can go to these maps and understand what these substitutions are likely doing with respect to these important phenotypes. So to do that, we've been leveraging a platform called yeast surface display. This is a classic platform, especially in fields like antibody engineering, where it enables us to take an amenable lab organism, yeast, and sort of make them our little biochemical factories. So we can have our yeast express an RBD of interest sort of tethered to their cell surface. And then when we have that RBD there in sort of extracellular environment, we can probe for different biochemical characteristics of this yeast displayed RBD. So for example, we can use fluorescence labeling of a C-terminal epitope tag to determine how efficiently different RBD variants are expressed on the cell surface, which is sort of a proxy for the folding and stability of an RBD variant. We can incubate our yeast with soluble ACE2 protein, for example, to determine how different RBD variants impact ACE2 binding affinities. Or we can incubate these yeast with monoclonal antibodies or polyclonal antibodies that target this RBD to determine how mutations might enable escape from binding by this antibody. Because this is sort of an extracellular assay, we can treat it like a traditional biochemical assay, for example, doing titrations of our extracellular ACE2 ligand. And since it's a fluorescence readout, for example, via flow cytometry, if we take the SARS-CoV-2 RBD on the surface of yeast and titrate in human ACE2 receptor, we can see the sort of titration of the fluorescence signal of ACE2 binding increase over time, which allows us to fit these sort of binding curves and derive the kind of dissociation constant, which is the measurement of the strength of binding of, in this case, SARS-CoV-2 RBD for the human ACE2 receptor. But of course, this isn't high throughput. This is a one-at-a-time assay. And so what we do to make this high throughput is obviously working within the context of variant libraries. And so rather than have a yeast population all expressing the SARS-CoV-2 RBD, we can make a population of yeast where each cell has some different genetic variant that they're containing and therefore expressing the protein of on their cell surface. On the next couple slides, I'll show you several different types of libraries we can use in this type of platform, but it's very flexible. What we then do is take this library, and the same way I showed a titration before, we can take this library now and incubate across a titration range of ACE2. But rather than just observe the shift in that kind of unimodal peak, for a single variant. Obviously here we have a mixed population of cells, which all have different affinities. And so you can kind of see that this flow curve has a lot of variation because there's different mutants in the library that impact this binding. And so what we do instead of just measuring fluorescence is we actually use FACS to actually partition cells based off of their ACE2 binding fluorescence signal. And then we can use deep sequencing to see which cells fall into which bin across the sort of titration series. And so, for example, what I'm showing here is if we have, for example, a high affinity variant like the light blue yeast cell, we can see via sequencing that it begins to titrate up, earlier than perhaps the green variant or the orange variant, which is not able to bind ACE2 at all, and so would get sorted into bin 1 down the entire titration series. And so when we deconvolve all of the sequencing data across all of these FACS bins, what it allows us to do is to basically fit variant-specific binding curves all in this one single experiment. So from one experiment, we can get thousands of independent dissociation constants and the curves underlying those dissociation constants, giving us these very precise measurements of affinity within these variant libraries. So I don't typically go into this, but since this is a DNA-centric crowd, I thought I would just give a little bit more details of how we make our libraries. And in particular, we've been leveraging the concept of barcoding libraries. And in particular, this has allowed us to do many different things and pool different things together in a very modular way. So a lot of what I'm going to talk about today is kind of our most standard experiment, which is where we're making site saturation mutagenesis libraries. So we take, for example, our SARS-CoV-2 RBD, and we just tile down each site in the RBD, making site saturation mutagenesis libraries containing all possible amino acid changes at that position. What we then do is take this oligo pool of mutant variants that we get, and we clone it into our yeast display vector along with a second DNA segment that we make in the lab, which contains this sort of randomized N16 barcode sequence. And so when we do this Gibson assembly, we're going to end up linking each single mutant in our library to some distribution of randomized N16 barcodes, which we can then use PacBio sequencing or any sort of long read sequencing paradigm to basically get long reads that span RBD to barcode, which allows us to create these tables that give us an association between which N16 sequence, right? ATT, CCC, GDG, whatever, is linked to what RBD variant, N331T. And so we get this big table of all of our barcodes linked to which mutant they're associated with, such that when we then take our library into yeast and do this FACS and especially the sequencing, it allows us to have a much simpler sequencing paradigm where we only need to sequence this N16 barcode and then just cross-reference to that lookup table to know that this barcode represented the orange cell genotype and this other barcode represented the light blue genotype, and so on and so forth. I wanted to show just a couple kind of general QC stats that this approach works really well. So when we order these SSVL libraries and then do this whole cloning paradigm, and then we get the PacBio data, we actually get to see everything that's in our library at high resolution. And we can see, for example, that we are getting very precisely the single mutants that we encode. So this is each of the variants in our library and how many amino acid mutations are present in that variant. You can see we only have a very small subset of kind of product higher order mutants. And we can also look at the kind of mutation rate across our RBD gene. So these are the 201 codons in our RBD, and showing the mutation rate. We can see that it's quite even. So we have good coverage of mutations in each position in the library without kind of unnecessary stop codons besides the ones that we encode is to have just a few negative controls, and without tons of extra synonymous mutations as well. So these are very efficient libraries. The other thing I wanted to mention is that because we do this PacBio association with barcode, we end up with a very modular analysis where we can take different libraries, which might have totally different design principles, but then are linked to barcodes, and we can pool different libraries together for different experiments as it becomes relevant. And I think at the end of the presentation, I'll show one example of that where we've pooled different library types together. So there's a lot of different things we can do. For example, rather than order, mutant libraries of our gene of interest, we can also just order a bunch of different genes of different homologs. For example, across that phylogeny of the SARS-related coronaviruses, we can just order each of those RBD genes, clone them into this platform, get the barcode associations, throw those then into the same experiment. Or we can create site saturation mutagenesis libraries across multiple related backgrounds, do the association, and then pool them all together for experimentation. And so this barcoding approach allows you to basically do experiments on anything that you can create genetically in the lab. All right, so let's get into some data and experiments. So what I was going to tell you about first was sort of the first experiment we did on SARS-CoV-2 RBD. And so this takes us back to, I think, February 2020, when we were all starting to pay attention to this coronavirus. And we knew that the RBD would be important to the virus, but really the entire field had no idea what the capacity for future evolution was going to be of this virus. There were a lot of debates at this time as to whether the virus was just going to sweep through the population and then run out of naive individuals and go away, or whether it would have capacity to evolve, for example, antigenically to reinfect individuals. And so we wanted to just get a basic understanding of what is sort of the mutational tolerance and the evolutionary potential of this key domain for this newly emerged virus. And so to do that, we created a demutational scanning library within the SARS-CoV-2 RBD, where we're taking each of the 201 sites and sampling each of the possible 19 amino acid mutations that are possible at a site, yielding about 4,000 mutations, which we then measured through that high throughput FACS sequencing platform that I showed before. So this is an example of what the data looks like out of one of these experiments, in this case, when we were measuring the impacts of each mutation on binding to human ACE2 receptor. So in this heat map, each column is one of the sites in the RBD from residue 331 to 531. Each row is one of the possible amino acid mutations that we can introduce, roughly grouped by sort of biochemical characteristics. So then each of these cells represents the measured effect of that particular mutation at that site on ACE2 binding, according to the scale bar here. So red are mutations that severely decrease ACE2 binding. White are mutations that don't really impact ACE2 binding affinity. They're neutral with respect to receptor binding. And blue are mutations that enhance ACE2 binding. And so maps like these are extremely rich, and obviously we can't get into all the different details here. But one thing I always like doing with these maps is going to a structure. You can really take each of these site-specific profiles and in the structure understand, why prolines are so bad in this stretch, and why, these polar residues are okay, whereas these non-polar ones are detrimental, so on and so forth. So here's an example, site 488, which is a cysteine in the wild type SARS-CoV-2 RBD, and we can see every single mutation of this site is highly deleterious, and that makes sense. This is a cysteine that forms a disulfide bridge, and none of the other amino acids can complement that characteristic of cysteine. But we also saw a ton of positions like this one here, site 493, where we saw a lot of variation in mutational effects. So sure, there's some mutations that decrease binding, but there's also a lot of mutations that are tolerated or even enhance ACE2 binding affinity. And so when we got these data, we didn't really know what it meant, besides that we could look at this and see there are a lot of tolerated mutations. There doesn't seem to be that much constraint in this key domain. But we had no idea what it was really going to mean, because there wasn't much genetic variation happening yet in SARS-CoV-2, but we had these measurements. And what that meant was that, fast forward to kind of fall of 2020, when the first variants of concern like alpha and beta and gamma began to emerge, or for example, later Omicron, they were sampling mutations from this map, such as N501Y here, where we could go to our map and see, okay, this mutation, it's the only RBD mutation in alpha, is enhancing ACE2 binding. That probably means something important. And I always like to joke that we were the first people to measure that N501Y was affinity enhancing, because we measured it before it evolved in the first place, right? Tons of other labs then quickly followed up after alpha evolved to make these measurements, but we already had it because we just sampled everything that was possible to begin with. One other pattern I like to point out here is that mutations like N501Y are not by any means unique in this landscape. So there's plenty of other ACE2 enhancing mutations present. And it's a little bit less obviously striking at first, but what ends up being really, really important for the evolution of this virus is the fact that there's all of this sort of white space in the landscape as well. So these are all the mutations that the RBD can sample without sacrificing its ability to bind ACE2 receptors. And this is what creates a large evolutionary capacity for immune escape, which I'll touch on briefly in the next couple of slides. I also like to add that this lack of constraint at many of these positions is not a simple consequence of these sites not mattering. So what I'm showing with the kind of black indicators above are the sites that are actually in direct structural contact with ACE2 in the crystal structure. So 5 angstroms or less away from ACE2. And we can see that plenty of these positions are the ones that have tons of mutational tolerance or even evolvability, really showing that the sites where all this tolerance that I'm talking about are the ones that actually matter for ACE2 binding, and they're not just far away from the interface and therefore unconstrained. So to kind of give an idea of what this tolerance to mutations has enabled, I was going to kind of start with a little vignette based off of the monoclonal antiviral antibodies that were being developed for clinical usage of COVID-19. And so at the very beginning of the pandemic, again, before we knew that this virus was going to evolve so readily, A lot of the companies that were developing very quickly the first monoclonal antibodies to use for therapeutic treatment of SARS-CoV-2 were premising their idea, their work on this idea that if they find the antibodies that target the ACE2 contact surface of the RBD, these antibodies should be resistant to viral escape, because the classic idea is that there's a lot of functional constraint at these residues. So again, this is showing the data from the last slide, our measurements of the effects of mutations in ACE2 binding mapped to the structure. And what I'm showing in spheres are those residues that are the ACE2 contact positions. And each site here is colored by the average effective mutations on ACE2 binding. So again, white are sites where there's not that much constraint, and then red are sites where mutations on average have quite detrimental effects on ACE2 binding. And so when we look at this idea about the ACE2 contact surface, we can see that, yes, there are some dark red, highly constrained ACE2 contact sites, but there's plenty of interspersed kind of white and pink residues, showing that there's plenty of vulnerabilities at the ACE2 contact surface as well, in terms of the ability of this virus to escape. And we were able to see that in some of the early data that was coming out. For example, this was a study done by Regeneron when they were kind of developing their monoclonal antibodies, where they took wild type SARS-CoV-2 virus and grew it in the presence of different monoclonal antibody candidates to look at what type of escape mutants come out from that selection. And we looked at what types of sites they were getting these mutations at. We could see that they were sub-selecting out the sites at this ACE2 contact surface that are the pink and the white, the tolerant sites, showing that, in this sort of mosaic surface of constraint and tolerability, it only takes one mutation at one of these tolerable residues to escape an antibody without sacrificing ACE2 binding. We did develop a kind of a secondary assay that helped us to see this in a little bit more concrete detail, where we use our GMS libraries, not for ACE2 binding, but to identify mutations in the RBD enabling escape from different monoclonal antibodies. And just because you can get a little flavor of what this data looks like, I'm showing here the data for an antibody, Regeneron 10933, which was one of the components of that therapeutic cocktail. And so what I'm showing here is how we measured escape from binding by this monoclonal antibody for mutations at each site in the RBD. So we see these peaks, which are the sites where mutations enable loss of binding for the antibody. We can zoom in on those peaks and look at them at the mutational level to see which individual amino acid residues at each site enable escape. So some of the most important sites for binding by this antibody, like F486, any mutation to that residue will abolish antibody binding, where some of the other sites have more restricted profiles where only certain amino acids are sufficient to enable escape from antibody binding. One thing we can do is take our sort of orthogonal measurements of ACE2 binding affinity and layer it on this map to identify which amino acid residues are tolerable versus constrained. So dark brown are mutations that have no impact on ACE2 binding, and so these are ones that presumably could easily occur without the virus sacrificing this other trait that it needs, whereas the ones that fade away to yellow are those that escape the antibody, but they also have severe deleterious consequences for the ability of the virus to get into cells. We can map our escape, so the height of these peaks, onto the RBD structure and kind of see that there's the semblance of a sort of epitope here, which of course maps to where we can know from crystal structures that the antibody binds. Although I do always like to point out that this kind of functional definition of the epitope gives more nuanced information, such as the fact that this F486 is really a hotspot for the interaction, and some of these sites where the antibody is contacting perhaps doesn't matter as much for interaction. And then last, we can kind of, for example, compare it to these viral escape selections I was showing earlier and see that indeed the viral escape selections are finding mutations that enable loss of antibody binding without sacrificing ACE2 binding. So they're these darker colors without sampling these more constrained mutations. So since those first studies on some of the therapeutic antibodies, we, and now especially Yunlong Chao's lab at Peking University, have performed this experiment for, at this point, hundreds of antibodies, mostly from Yunlong's group. And what that allows us to do is basically simulate what escape might look like from a random selection of antibodies, such as those that are polyclonally developed in our own bodies. And so this is a tool that Jesse developed that kind of aggregates all of these experiments to determine which are the sites that across many independent antibodies are the biggest hotspots of escape. And so I'm labeling some of the strongest peaks here, which for those who have been paying attention to SARS-CoV-2 evolution, you might already recognize. I guess I'll show this first. And so another kind of example of why tolerance to mutation has enabled antibody escape is the fact that these sites that have the strongest antibody escape often don't have much constraint for ACE2 binding. So again, this is this average effective ACE2 binding. These are sites that are highly constrained. These are sites that can mutate without losing cell entry. And we can see that a lot of these strongest escape sites are indeed those that can mutate just fine. And so then it's kind of no surprise that in addition to mutations like N501Y, which are important for ACE2 binding, this virus has also easily been able to sample mutations that enable immune escape. So for example, many of the early variants were sampling this E484 mutation, K417 mutations, and then of course, Omicron wiped through basically fixing a mutation from each of these peaks, basically wiping out all of our immune responses from early viral exposures. And that's really been facilitated by the fact that these mutations can occur without suffering these large consequences for ACE2 binding. So what I want to get into next is, trying to find a clock here, was as these variants of concern began to diversify, a lot of people were wondering, what does it mean? Besides the kind of focal impact of these mutations on ACE2 binding and immune escape, what does this mean for us and what does it mean for subsequent viral evolution? So my earlier data was presented from the ancestral Wuhan 1 strain, but all of these variants have been fixing mutations which could impact the subsequent effects of future mutations. So again, as I showed here, N501Y is affinity enhancing here, but as these backgrounds begin to change, we wanted to know how do the effects of the rest of these mutations change across backgrounds. So this is this concept of epistasis, which is a general concept that refers to the non-additivity of mutational effects. The way I'll be showing epistasis is through the idea that we'll be identifying when do mutations have different effects if they're introduced into different sequence backgrounds. So if we introduce a mutation in the SARS-CoV-2 ancestral strain versus Omicron, and it has a fundamentally different effect on ACE2 binding in those two backgrounds, that's indicative of epistasis. We know that from other work that epistasis can be important in evolution over longer timescales of these SARS-related coronaviruses. So I'm showing a mutation here, N501Y, which, as I've shown from the heat map in SARS-CoV-2, is affinity enhancing. So this is a binding curve, wild type. The left shift of this curve indicates affinity enhancement of the N501Y mutation in this particular sequence background. But when we introduce the same mutation in most other SARS-related coronaviruses, such as SARS-CoV-1, the same mutation is actually severely affinity decreasing, showing that this N501Y mutation has a lot of context dependence in its affinity enhancing effect, or epistasis. So to see whether this was occurring over kind of the smaller timescale of SARS-CoV-2 evolution, right? These are different at about 25% of RBD residues. We basically took our computational scanning approach, and we now just did it in parallel across different RBD variant backgrounds that fix the substitutions that define these variants. So we have the same data as I showed before in the Wuhan 1 background. Now we fix that N501Y mutation and make another library and measure all the impacts of mutations on the backdrop of N501Y or on the backdrop of L452, T478K, and so on. And so now we end up with these even bigger heat maps, which are even harder to visualize. But just as a kind of teaser, you can start to see some interesting patterns. So for example, there's a mutation here that is white, so it doesn't really impact ACE2 binding in the Wuhan background or in Delta and Eta, whereas it's dark blue in the Alpha and Beta background. What's that about? Or we can see kind of stretches of residues that, for example, are darker red in one background, indicating more functional constraint, whereas in some of these other backgrounds, that constraint is relaxed slightly. So these mutations have a less deleterious effect in these other backgrounds. So obviously, visually from the heat maps, these patterns are very hard to distinguish. And so we wanted to develop a metric that allowed us to more clearly identify these so-called epistatic shifts in mutational effects. And so here I'm showing two different sites to kind of illustrate what this metric is capturing. In these scatter plots, what I'm doing is showing the affinities for ACE2 that we measure of mutations when they're introduced, for example, in the beta background that fixes 3 substitutions relative to the effect in the ancestral Wuhan background. So in these scatter plots, each of these letters is an amino acid mutation, and we're showing the affinity of that mutant variant in each background. So site 484 is a site that doesn't have a strong epistatic shift. And we can see that that's because these 20 amino acids are basically on the 1-to-1 line here, indicating that mutation E484F has the same biochemical impact on ACE2 binding, regardless of which of these two backgrounds you introduce it into. And that's in contrast to site 498, for example, where we see a lot of scatter off of this line. What the scatter shows is that mutations like Q498, W, H, or Y are actually affinity enhancing in the Wuhan background. So this is the wild type affinity. To the right means higher affinity, but they're below the line for the beta background. So they decrease affinity in beta. or vice versa. This R mutation is slightly affinity decreasing in Wuhan1, but it has a strong affinity enhancing effect in the beta background. So this is just two sites. Obviously, we have 201 residues in the RBD, and we have many different variant backgrounds. And so I'm showing here is this epistatic shift compared to the ancestral background in each of these different variants where we did these measurements. And so we can see that mutants like, or variants like Delta and Eta have relatively modest epistatic shifts when we compare it to what we saw in the alpha and beta backgrounds. And in particular, the lines for alpha and beta are basically identical. And we think that's because this focal N501Y mutation that's shared between these two variants is a really big epistatic perturber of this landscape. We can see that, for example, structurally. So here is the RBD colored by this epistatic shift metric. And I'm showing where N501Y occurs on the structure here. And so we see that the dark blue residues and the other residues with modest epistatic shifts are really emanating from this focal N501Y substitution. I do always like to point out, though, that it's not obvious from structure alone that this mutation was going to have such a strong perturbation effect on the impacts of mutations at these sites. For example, structurally, if we look at the backbones of different variant RBD structures that have been solved, we really see no change to the backbone, for example, around the N501Y mutation. And even when we zoom into side chains within these static crystal structures, there's nothing obvious that shows or would have allowed us to predict that N501Y was going to change the effects of mutations at these other sites so dramatically. And yet we've seen that these epistatic shifts have mattered quite a bit for SARS-CoV-2 evolution. And the kind of best example of that is this epistatic interaction between N501Y and a second mutation, Q498R. And this was something that was described, for example, in experimental evolution studies as well. In our data, what we can see is in this kind of plot I'm going to show you here is we can look at the affinity of different mutant combinations to kind of visualize what this epistasis means. So here is the affinity of Wuhan1, as I've shown you many times before. N501Y by itself is affinity enhancing. And by itself, this Q498R mutation is slightly affinity decreasing. So if these two mutations didn't interact epistatically, we'd expect the double mutation to have affinity here, right? This Q498R would have the same impact on this background, and that would make this little parallelogram structure. But in reality, when we have the affinity measured for this double mutant, we see that its affinity is much greater than you'd expect from the kind of sum of the individual mutations. And so the difference between this expectation and the reality is how much epistasis occurs between these two substitutions, which allows them together to give very, very high affinity increase. So when we first saw these data, we're like, oh, that's really strong and very striking. I wonder what it means. But we were pretty perplexed because when we went to SARS-CoV-2 evolution, we didn't see these mutations occurring together very frequently. But it took just about 1 month until something happened that then did make sense, which was that Omicron emerged. And when Omicron emerged, it fixed 15 different mutations in its RBD, 2 of which was this double mutant pair. What we were able to see was that this double mutant pair had a really important role in Omicron emergence by basically buffering the impacts of mutations with respect to ACE2 binding, allowing Omicron to take such a large immune escape or antigenic jump. So to show that, what I'm going to kind of walk you through here is this plot where we're looking at basically the predicted cumulative ACE2 binding energy of a variant relative to Wuhan1, as we basically just add up the mutations, the 15 mutations that occurred in Omicron BA1. So I'm not saying this is the order that the substitutions actually occurred in during evolutionary emergence of Omicron, but rather just to illustrate basically the affinity gains and the affinity decreases that are happening amongst these 15 different substitutions that define the Omicron variant. So the kind of overarching picture is that the actual measured Omicron affinity, as well as the kind of predicted affinity we would give Omicron based off of our signal mutant measurements, is that this variant does have pretty similar affinity to the ancestral Wuhan1 strain itself, just a slight affinity enhancement. But this slight affinity enhancement is the result of a massive sort of addition and subtraction of affinity amongst these 15 different mutations, of which N501Y and the epistatic interaction with Q498R is a huge contributor to the kind of height of this arc. What we also noticed was that all of these mutations that are these little cuts back at ACE2 binding are enriched for being antigenic escape or antibody escape mutations, shown in red. I can show that this way here as well, which is taking our mutational measurements and looking at those that are affinity enhancing versus affinity decreasing compared to immune escape. So to the right are mutations that have stronger escape from antibody-based immunity. And what we can see is that the mutations that are enhancing affinity have hardly any effect on immune escape, whereas the mutations that are enabling immune escape all have these slight deficits in ACE2 binding. So what we think happens is that this virus needs to maintain a certain level of ACE2 binding to remain infectious. And when it wants to escape immunity, it typically can do that, right? No single one of these mutations has such a strong deficit that it would be inaccessible to the virus, but it does limit how many epitopes the virus can mutate all at once. And we think that the larger affinity buffer that exists that allows the virus to take more cuts at different antibody binding sites to escape more antibodies all at once. And that's fundamentally what underlied Omicron's ability to sweep through populations regardless of immune status, because it was able to fix all of these different mutations and all these different epitopes because of the affinity buffer that this epistatic interaction conferred. The other thing I'll show is that this is one key interaction that mattered for one focal emergence of a variant of concern. But we also see that kind of under the bubble of all this genomic surveillance of SARS-CoV-2, these epistatic shifts are changing what's happening over the course of SARS-CoV-2 evolution. So for this, we were able to take advantage of this great resource of tens of millions of SARS-CoV-2 genomes around the world, as well as people who collate all of that data and create tools like this global phylogeny called Usher that collates all of the sequences available into a phylogenetic structure. And so working with Will Hannon and Jesse's lab, what we did was just take this phylogeny and count how often every possible substitution in the RBD occurred, but basically partition it as to whether that substitution occurred on the backdrop of N501 versus the mutated Y501 background. So that's what I'm showing here is from the genomic surveillance data, whether a substitution was occurring more frequently in conjunction with the Y501 mutation versus whether it was occurring more frequently on the ancestral N501 background. And comparing that to our epistatic mapping from our deep mutational scans, where we can see mutations that have higher affinity on the Y501 backgrounds going up versus those that have better affinity when introduced into N501 backgrounds going down. And so what we can see is that, in addition to mutations like Q498R, there's all these other mutations that perhaps haven't fixed in a variant of concern, and yet they are still occurring at a higher background rate when they accompany this epistatic modifying N501Y mutation, whereas these are mutations that are occurring less frequently once Y501 genomes began to proliferate because they have worse impacts on binding in that background. And so I think this shows that this epistatic perturbation is, besides the emergence of these big clades of successful variants, is also impacting the occurrence of substitutions that are basically the raw material from which these variants sample. All right. I'll spend a couple quick minutes on some of the more recent work we've been doing. So that first study was all obviously variant background that were all pre-Omicron. Since then, we continue to just churn through different SARS-CoV-2 variants, repeating this deep mutational scan. So now we have deep mutational scanning data in the Omicron BA1, the BA2 background, and in a more recent preprint in the BQ11 and the XBB15 variant. And we are seeing that epistatic drift, as we're calling it, continues to proceed, although not as dramatically as was explained with that N501Y substitution. So here I'm showing that epistatic shift metric again, in this case versus Omicron BA2, and Wuhan 1 versus BA2 is basically dominated again by the N501Y distinction, where we see these strong epistatic shifts, whereas some of the more recent variants have much more muted patterns of epistatic shifts. There's not these huge peaks of mutations that have effects going all over the place. That being said, we are still seeing these kind of more mild epistatic shifts and drifts are still impacting viral evolution. So we were able to describe, for example, in XBB15 that there's epistasis between this Q493R mutation and its reversion with mutations at sites like 455 and 456. And it's these sites that are kind of the focus of SARS-CoV-2 evolution right now. So what's currently kind of replacing XBB15 or has basically replaced XBB15 in many locations is a variant called EG5, which is XBB15 plus this 456L mutation, which has been kind of epistatically changing in its effect over time. And then subsequent to that, there's also mutations at L455 that are beginning to occur, which is another site whose kind of effects of mutations have been changing over time, showing that kind of SARS-CoV-2 evolution continues to kind of pick up on mutations as they become available with this background changing of mutational effects over time. I'll sort of kind of basically wrap up here with kind of an overview of the different DMS experiments we've now done across different SARS-CoV-2 variants and what they're showing us. So what I'm showing here is basically a layout of the different backgrounds in which we've done these demutational scans in this kind of multidimensional scaling, which is similar to like T-SNE and things that UMAP that people use to like show relationships between data in, for example, single cell RNA sequencing data. So in this kind of map, backgrounds that are closer together had more similar mutational measurements in our demutational scanning data compared to variants that are further away in space, where there was more apathetic shifts in mutational effects. And when we look at the variants here, one thing we noticed is that the distance on this map really doesn't have a relationship with the genetic distance of variants. So here's the same layout measured by indicating how many substitutions distinguish each of these backgrounds from Wuhan 1. And we see that some backgrounds that are close are a single mutation away, but also alpha, which is really far away, is also a single mutation away, basically as far away as SARS-CoV-1, which is 52 substitutions away, showing that it's not just number of sequence changes itself that determines perturbations and divergence in this landscape, but rather it's particular substitutions that have large effects. So of course, N501Y is one of the biggest kind of distinguishers between the cluster up here and those that are down here. Another thing this thing shows is that even within the Y501 genomes themselves, there's kind of epigenetic drift over time, as I was showing, where, for example, the more recent variants have some of these sites that are differing more dramatically than they were, for example, in Omicron BA1 or BA2. The other thing that I didn't talk a whole lot about is, for example, SARS-CoV-1 is kind of in a different branch of space in this sequence landscape. So SARS-CoV-1 has T501, which is similar to N, but what it has that makes it quite distinct from all the SARS-CoV-2 backgrounds is Y498. And if you remember, 498 and 501 have a lot of really important interactions. And I thought this was interesting because one of the things that we noticed in our first demutational scanning map in Wuhan 1, when I pointed out all those possible blue mutations, affinity enhancing mutations, either Y498 or Y501 were affinity enhancing, but the problem is that they are not affinity enhancing together. So you can't have both of these mutations. And so it's almost like the ancestral SARS-CoV-2 strain in the beginning had sort of a fork in the road where it could have fixed Y498 or Y501, but it ended up going down this path, and that's kind of dominated SARS-CoV-2's evolution ever since. All right, so I'm going to wrap up. I had one shortened section that I wanted to show about the broader evolution of SARS-related coronaviruses, but I'll skip it. But I'll just kind of highlight that using the same platform for mutational measurements, as I was saying during the kind of library generation stage, we can also make libraries of different genes. So we can just order all the different RBDs from all the bat coronaviruses that span this phylogeny and put them into our platform to determine how all of these different bat coronavirus RBDs impact human ACE2 binding or binding to ACE2 from different host species. So with that, I'll kind of wrap up, and I'll thank the members of my new lab at the University of Utah. But I'd also like to thank Jesse, my postdoc mentor, which is where most of the work I talked about was performed. Lots of great collaborators. I'll just throw a plug in that we're in Salt Lake City now. So I started my lab at the University of Utah about 1.5 years ago, and we're just nestled right here on the kind of edge of town, right along the foothills of the mountains, and it's very nice and beautiful. And so if you're in town, come visit us. So thanks very much. I'm happy to take any questions, and thanks.
Thank you, Dr. Starr, for a great presentation. It's always exciting to learn how our partners are leveraging our products. We are now going to open it up for questions. So if you have a question, please make sure to ask it in the Q&A box. So our first question, does the yeast expression system glycosylate the RBD? If so, does that affect the binding affinity?
Yeah, that's a good question. So yeast do glycosylate at the same motif as mammalian cells, but the glycans are high mannose type, so they're not going to have the same complexity as many of the glycans that would be added with mammalian expression. One thing that kind of helps us in the context of these Sarbeco RBDs is that the glycans on the RBD, so SARS-CoV-2 has 2 of them, and they're both kind of projecting out here on the sort of side of the RBD. And so they're not really involved in ACE2 binding per se. They are important in the rest of spike. For example, they control the dynamics with which RBDs open and close. But when we're just taking RBD out just to measure affinities per se, they're not really getting in the way of that. There are some antibodies that bind to the surface and have to engage or tolerate the glycan. And we've seen, I guess, variable degrees to which the yeast display data recapitulates what happens with proper glycosylation or not. And so I think that's where sometimes we can get problems. So, we have to be aware that the glycans are different, but at least there is a glycan there. And we do know that, like in the mammalian platform, those glycans need to be there for stability of the RBD, even when it's the yeast glycan.
Thank you for that. So next question, it appears that the Omicron was reading your papers and fixing RBD mutations. Great work. Wonder what compensatory mutations were tolerated, fixed within the spike trimer to accommodate the RBD immune evasive mutations.
Yeah, that's funny. Yeah, I mean, evolution, that's a kind of a bigger picture, philosophical picture. That's been one of the most interesting things here is like, when we got our first mutational scanning map, we knew what it meant biochemically, but we didn't know what it was going to mean evolutionarily. And so it's been very interesting to see evolution tells us what does matter. And our maps actually do explain a lot, which, as a scientist, you kind of always doubt your own data. That's kind of the practice. So it's kind of funny to think about it that way. The question about compensatory mutations elsewhere in spike. I mean, so Omicron has the same affinity as Wuhan 1. So from that perspective, the compensatory mutations probably were that double mutant I talked about that offset the deleterious effects on ACE2 binding per se of the rest of those mutations. But that being said, like I said in the first question as well, there's definitely other dynamic things going on as well. So some of the RBD mutations that define Omicron have been suggested to impact, for example, RBD up-down dynamics as well. And there's other substitutions in the spike that are impacting, for example, fusogenicity and the efficiency of furin cleavage and all these other things. So of course, there's many other phenotypes impacting transmissibility and things like that, whereas we're just focused, on that biochemical interaction of RBDs and ACE2s.
How constant is human ACE2 interface sequence?
Yeah, so that's an interesting question. So it's pretty constant. There's no like high frequency ACE2 variants that have been shown to impact SARS-CoV-2 spike binding. But what I did have to skip, but is interesting, is that even though there's not tons of polymorphism in humans, within the natural bat reservoirs that host these viruses, there's tons of polymorphism within the ACE2s that's all localized to the interface right where these RBDs bind. And so that's what we're beginning to work on a lot more, kind of zooming out from SARS-CoV-2 itself to understand why is this arms race between bat ACE2s and the sarbecovirus RBDs driving this promiscuity that enables human ACE2 binding and spillover.
Not a question, but many thanks for a great presentation.
Thanks.
So next one, thoughts on measuring the effect of mutations on RBD protein expression during these DMS experiments? Is it a relevant parameter?
Yeah, I meant to mention that because I don't have a slide on it, but we do always also do the deep mutational scan for surface expression levels. And especially in the first study that we did, we found a lot of stabilizing mutations that actually ended up being pretty interesting. And we were able to work with collaborators at the Institute of Protein Design at the University of Washington who were developing or are developing still and have approved in parts of the world an RBD-based vaccine immunogen. And we were able to show that these stabilizing mutations that we got from our mutational scan when put into mammalian expressed proteins or even this vaccine immunogen, stabilize the protein, you get like 5-fold higher yields, better shelf stability and things like that. So that was really cool. And it's kind of complicated, but the mechanism by which these stabilizing mutations work, I've always thought is really cool as well, which is that in the full spike context, it's a trimer. And so there's 3 RBDs kind of packed in right here. There's actually a lipid binding pocket that bridges 2 RBDs. So the head of the lipid binds 1 RBD, and then a hydrophobic tail inserts deep into the core of a neighboring RBD. And what that means is when you take these coronavirus RBDs out from that context, there's now this hydrophobic void in their core because they're not binding the lipids anymore. And so what we were finding was that space-filling mutations in the core refill that pocket and greatly stabilize the RBD because of the loss of this lipid binding when you take RBD out from the spike context. And I've always thought that was kind of a cool thing.
Thank you. Next question, should we be afraid of bats virus mutation in the future?
I mean, we should be prepared for many different viruses that circulate in bats and rodents and other animal reservoirs. In general, there's tons of viruses out there that are diversifying all over the place. And I think SARS-CoV-2 was kind of a nice example, well, not very nice, but an example of how kind of blinders about evolutionary diversity out there in animal viruses can kind of mislead us. So what I had to skip was this slide here, for example, where before SARS-CoV-2 emergence, this is what we knew about viruses. We knew SARS-CoV-1 and a bunch of related bat viruses, which all bound ACE2. But the viruses in Africa and Europe didn't seem to bind ACE2, at least human ACE2 as had been described thus far. And this other clade of bat viruses didn't bind ACE2. So people were really focused on SARS-CoV-1 related viruses because that's where they thought the spillover concern was. And then flash forward to 2020, and this branch that wasn't really known about or wasn't very focused on emerges with SARS-CoV-2. And obviously SARS-CoV-2's potential for pandemic spread is unprecedented. And so if we, only look at what we know about among bat diversity, we're missing a lot of other viruses that we're not seeing. And I would say if you're going to be developing monoclonal antibodies for pandemic prevention or like pan-Sarbecavirus vaccines, you need to know what the breadth of viruses is that you should be prepared for, because the spillover isn't going to be likely the virus we already knew about. It's probably going to be something that's evolved in the meantime that we just hadn't seen yet.
Thank you. The next question, which mutations should we be targeting to monitor future variants via multiplex RT-PCR?
I would say what appears to be the strongest driver or the most predictable driver of like what mutation occurs next, at least within the RBD, has been pretty well modeled by like immune escape phenotypes. So there's, this is the RBD, there's many different places where antibodies can bind, and we have all these experiments now that have began to unveil where these antibodies bind across the RBD. And what happens is we have immunodominance. So most of our bodies to the ancestral strain made a ton of antibodies to E484, and that was where the most pressure was on the virus, and so that was the first antigenic mutation it took. But once E484 occurred, the second best class of antibodies was then the major pressure for the virus, and so then it started fixing K417 mutations, and then Omicron was having these 444 mutations. So that tool that I had a link to that Jesse's made, this antibody escape calculator, allows you to simulate, okay, once all of these epitopes are mutated, where's the pressure going to shift next? And that has actually worked pretty well for predicting what's next. For example, it made it pretty clear that F456L was going to be the next mutation after XBB.1.5, and then EG5, of course, is the one that did emerge. So that's kind of the tool I think that has proven the most predictive. That's coming from someone who's pretty pessimistic about true forward prediction abilities. So,
thank you. How well known are the ACE2 sequences for various species of bats? There are so many bat species. Are there repositories for bat genomics?
Yeah, so there actually is a newly emerging bat repository for bat genomes. They're calling it, I think, 1,000 bat, is it 1,000 genomes project is the human one, so they're calling it 1,000 bat genomes project. And that's, I think it's a relatively new thing. So I think a lot of their data hasn't been published yet, but it's like a community resource. And so if you're in the field, you can, I haven't subscribed to it yet, but you can like get access to the data as long as you satisfy certain citation and waiting on publishing and things like that. And so we're getting a lot more bat genomes for sure. In terms of how many ACE2 sequences there are, I guess I have a little bit of blinders with respect to a particular genus of bats. So it's the rhinolophus bats, the horseshoe bats, that basically all the Sarbecaviruses have been sampled from. And there's, I don't even know, there's probably 50 to 100 species of those, and maybe 20 of the species have ACE2s. But I would say the biggest issue is that for species for which multiple individuals have been sampled, they keep getting different ACE2s. So there's also tons of ACE2 variation within species. So if we really want to know all the possible ACE2s that are out there, we would have to take really extensive sampling of multiple bat species simultaneously to get all the polymorphisms. So I think that's all I'd say. I think there's tons of ACE2 variation, especially within the rhinolophus species, that there would be a lot of value in learning more about, I think.
Mm. So one of the last questions. So once you've identified the impact of these individual amino acid mutations, would the next step be to look at these key mutations together using something like a combinatorial library?
Mm-hmm. Yeah, I think, as everyone knows, the balance between single mutants and combinations is always a hard one to strike. There are some groups who have taken more of that approach. So take the variety of, what's the name of the PI? They're at ETH Zurich. I forget the name of the group, but they've taken, for example, our single mutant scans and said, what are the mutations that are generally tolerant? Can we make combinatorial libraries within that to project further into sequence space? So people are definitely doing that. And especially if we want to study, like now that if we want to study epistasis. So now, we've started to see here are some of the really important epistatic modifiers like N51Y and Q493R. we could start making combinatorial libraries focused on those to get, because the inherent problem with epistasis is it makes evolution unpredictable, because epistasis means something does, like a mutation has an effect that's different than you expect, and you can't know that unless you measure it. And so that's what limits single mutant scans in general, is that you don't know if a mutation is going to have a different effect in a different background, not that far away from where you measured it. And so we could take an approach of kind of combinations focused on the places where we think there is epistasis to maybe get a little bit more projection into space, without maybe perhaps worrying about other sites where we don't think there's as much epistasis, where our single mutation measurements should be robust, at least for a while.
Got it. So it looks like we still have a few more questions rolling in, but maybe we'll take one more. Outstanding talk. Will you apply your DMS approaches to address protein-protein interactions that affect other emerging viral diseases?
Yeah, definitely. So, as I started to show, we did a ton of work on SARS-CoV-2. We're now expanding to the broader family of Sarbecoviruses, but we're beginning to set up the same series of things, especially in other coronavirus families. So one of the first ones we're looking at is MERS coronavirus and its related bat Mirbecoviruses, which have a lot of variation themselves. And again, MERS-CoV is what everyone's looking at, but that's not to say that MERS-CoV-2 isn't going to be from one of these other families that just hasn't been paid much attention to yet. And in general, like other coronavirus subgenera, of which there are many, seem to work particularly well in the yeast display platform because of this modular RBD. And then we are doing some work on HIV, which is actually what I had started on my postdoc, was studying HIV antibody kind of coevolution with diversifying within host viral diversity. And so we are also working a little bit more on that kind of HIV antibody coevolutionary stuff as well. And that's the short-term stuff. Yeah, so we're beginning to work on kind of other systems as well.
So I think with that, we're going to end the Q&A session. For the questions that we didn't get to, we'll send them over to Dr. Starr, and then we'll send over his response. I do want to end with a comment from one of our attendees. Thank you for the great work and good luck in the future. So with that, thank you, Dr. Starr, for a wonderful presentation. It's always enlightening to learn, and hear how our partners are using our products. Just a quick reminder for everyone who's here, after this webinar, you'll be redirected to a brief survey. So if you have 1 minute to spare, we would really appreciate your feedback. Other than that, thank you everyone for coming and have a great day. Thank you.
Thank you all.